EPFL engineers have developed a neural interface that disappears harmlessly in the body after several months and allows natural tissue to grow back. What is more, it can be implanted in a patient's blood vessel rather than inside the brain, thereby avoiding the need for invasive surgery. Some implants, like pacemakers, can last for years, while others wear out quickly due to technical weaknesses. New neuroprosthetic devices are thinner, more flexible, and more elastic, making them better suited to the mechanical properties of brain tissue. But that also makes them more fragile and less durable. Removing these implants can be difficult or even impossible because it generally requires invasive surgery. Engineers from EPFL are developing a new generation of biodegradable implants that get around this problem. The first obstacle they faced relates to the short lifetimes of biodegradable materials, which limit how extensively they can be used. The biodegradable neural interfaces currently being developed only work for a few days. That severely restricts the range of potential applications. The neural interface developed by the EPFL team is made entirely of polymers that deteriorate naturally after several months. It can therefore be used in medium to long-term applications such as monitoring epileptic activity or supporting neurorehabilitation after an injury. The engineers also found that the surrounding tissue grows back naturally once the implant has disappeared. That is different from most implants, which are inert and don't interact with the tissue. The team also wanted to create a device that could be implanted without requiring a major operation like craniotomy. Conventional neural interfaces are placed directly in the brain, and the tiny electrodes they contain stimulate or record cerebral activity. These devices are used to treat Parkinson's disease, epilepsy, and compulsive disorders, for example, and to study the brain. The implant developed by the EPFL researchers eliminates the need for invasive surgery as it can be implanted in a patient's blood vessel. The researcher says that they modeled their implant after stents, which are used to widen arteries and veins. The surgical procedure has become fairly routine and the recovery time is short. EPFL's neural interface can communicate with the brain but doesn't have to be in direct contact with the nervous system. Because it is made out of polymers rather than metal, it tends not to provoke a strong inflammatory reaction. The team's findings have been published in the journals Biomaterials and Advanced Materials Technologies. The researchers says that their research shows it is possible to develop minimally invasive neuroprostheses that interact with the surrounding tissue. This opens up new possibilities for applications in neurotechnology and expands the range of patients who can benefit from them.